This is the part one for the Metabolic Pathways chapter recorded lectures. And this is actually chapter 6. I did not, you may have noticed, I did not record a lecture for the cell chapters, both of the cell chapters. I've been sick and I apologize for that, but I definitely wanted to do a recorded lecture of this chapter because it's actually got a lot of chemistry in it. And in the past, I have taught this chapter broken up into 2 chapters. I've taught it as photosynthesis and then a separate chapter on cellular respiration. So I did want to record this one so that you could watch the recorded lectures and maybe that would help you understand the material in the chapter. So we're starting off with catabolic reactions. Anytime you hear the word catabolism or catabolic reactions, these are breakdown reactions. So the reactions which break down the food that you eat into smaller molecules are catabolic reactions. And one thing about catabolic reactions is that when they break the chemical bonds in the larger molecules and break them down into smaller molecules, they release energy. So one of the examples is during cellular respiration, the energy contained in the bonds of glucose, because glucose, even though it's a simple sugar, its chemical formula is C6H12O6. So it's got a total of 24 atoms in it, and those atoms are chemically bonded together. And those chemical bonds store potential energy. So when those chemical bonds are broken, that energy is released in small bursts, and some of it can actually be converted to adenosine triphosphate or ATP. And that is the molecule that is used for energy in cells. Most of the energy from glucose is lost as heat, but enough of it is captured so that it can keep the cell running. It actually is pretty energy efficient. It's somewhere around 39% energy efficient. So that would mean that only 61% would be lost as heat. And that's actually very energy efficient for a machine or a process, a chemical process. Okay, what you're looking at here is the inner and outer membrane of a mitochondrion. Now, if you remember in your cell chapter, the mitochondrion kind of looks like this. It's got an outer membrane and then it's got a folded inner membrane. And so it's not showing you that inner membrane folded here, but both of the membranes are composed, as you can see here, of a double layer of phospholipids, a phospholipid bilayer. And the reaction of cellular respiration occurs mostly inside the mitochondrion in our cells. The first reaction for cellular respiration, which is called glycolysis, occurs in the cytoplasm, but the other reactions occur in the mitochondrion. And the final reaction, in fact, what you're looking at here is a process called the citric acid cycle or the Krebs cycle. That occurs on the inside of the inner membrane of the mitochondrion. And that does produce some ATP, but not very much. But what it really produces are electron carrier molecules that we will learn about. They're called NADH and FADH2. These molecules provide electrons and hydrogen ions to the final step of cellular respiration, which is called the electron transport chain. And the process is called oxidative phosphorylation. Now, we will go over this process in much more detail, but this is just an overview to let you know that basically this is the reaction that we're going to learn about that provides us with ATP from glucose from the food that we eat. We will be in photosynthesis and cellular respiration. We'll be dealing with oxidation reduction reactions or redox. And all you really need in chemistry, you would go into much more detail, but all you really need to know in biology is what it means for a chemical to be oxidized and what it means for a chemical to be reduced. And I learned this years ago, this little mnemonic device, oil rig, and what that stands for is oxidation is loss of electrons and reduction is gain of electrons. So when a molecule is oxidized, it has lost or it loses. That molecule loses electrons. When a molecule is reduced, that molecule gains electrons. And so that's going to come up in this chapter is redox reactions. This page, okay, it says some redox reactions change the amount of electron density in a particular atom by altering how it shares electrons in covalent bonds. Below, carbon has lost electron density because oxygen is now hogging its electrons, while oxygen has gained electron density because it can now hog electrons shared with other elements. This is a very complicated way to say that this molecule, which contains many hydrogen atoms, is the one that loses electrons because a hydrogen atom is actually composed of, one proton and one electron. So that proton is often called just a hydrogen ion. So a hydrogen atom that loses an electron is a hydrogen ion. And this is, anytime you see hydrogen in a chemical, that means that it provides electrons and hydrogen ions to other chemicals. So this molecule right here would be the molecule that is oxidized. And it also makes sense because oxygen is being added to it. This is oxygen gas. So it sounds like that the C4H10 is the molecule that will be oxidized. So what is happening is all these hydrogen atoms are being given to the oxygen to make water, and the carbon molecule is being oxidized into carbon dioxide. So it's losing electrons. So the carbon molecule, we'll call it molecule A, is oxidized. Oxygen, which is molecule B, is reduced. And this is what they look like when the carbon molecule is oxidized, it becomes carbon dioxide. And when the oxygen is reduced, it becomes water. So reduced means it's going to take some of the hydrogen atoms, it's going to gain some of the hydrogen atoms. Okay, so energy in redox reactions. Glucose contains lots of energy. This is the structural formula for glucose. We already looked at the chemical formula, C6H12O6. Glucose is eventually broken down into carbon dioxide and water. So basically the redox reaction that does that is called cellular respiration. And this is just basically saying all these hydrogen atoms are sources of hydrogen ions and electrons. So glucose contains all these electrons that it ends up giving away to form water. The hydrogen ends up forming water, forming H2O. And so glucose is the, since glucose loses electrons, glucose is oxidized, but you would call it the reducing agent, and it forms carbon dioxide whenever it loses those hydrogen atoms, which contain electrons. So glucose by losing hydrogen, it's losing electrons, and loss of electrons means oxidation is lost. So glucose is oxidized because throughout the processes of cellular respiration, and there are many, many chemical reactions that occur during cellular respiration, but during those chemical reactions, glucose is slowly oxidized. It slowly loses all of its hydrogens and eventually becomes carbon dioxide, which contains no hydrogen atoms. And just remember that when a chemical contains hydrogen atoms, that the hydrogen is the source of the electrons. We have coenzymes, and we've talked about them already, but the coenzymes that serve as oxidizing agents, meaning they are sources of hydrogen ions and electrons. One is NADH. You don't have to know the name. I'm not even sure that I could say it. Nicotinamide adenine I think it's dinucleotide. But anyway, you don't have to know it. I obviously don't know it very well. But you do need to know that the symbol for it is NADH. And NADH provides hydrogen ions and electrons through that hydrogen atom. And by doing that, the NADH becomes oxidized and it reduces, another chemical by helping it to gain electrons. And that gain of electrons can ultimately be used to form ATP. And ATP is the energy currency for our cells. That means just like we use money to buy things, we use ATP for energy. It's like we use ATP to buy energy. ATP, the more ATP we have, the more energy that our cells have to carry out the processes of living. Now, there are two ways ATP can be produced. One way that ATP can be, I'm so sorry. This is actually the breakdown of ATP. This is showing you how ATP is used to give energy to a molecule. I'm not sure we need to go over this. Energy derived from glucose is used to convert ADP into ATP. When ATP is used in a reaction, the third phosphate is temporarily attached to a substrate in a process called phosphorylation. So this reaction is showing the breakdown of ATP, but what we're more concerned with, and what I wish they would have shown, is if you can picture this in reverse. Okay, so this chemical is just some chemical. This the helix represents some large chemical, and then the purple P represents a phosphate group. So the green chemical is called the substrate, and it contains a phosphate attached to it. So what happens is it donates its phosphate to ADP, and then ADP, which is adenosine diphosphate, gains this phosphate and becomes adenosine triphosphate. So picture this in reverse, and then the substrate has lost its phosphate. So here. So if you can picture the reaction in reverse, that is substrate level phosphorylation. That is how ATP is produced. Basically just taking a phosphate from some chemical and adding it to ADP, forming ATP. So that's called substrate level phosphorylation. And some of the ATP in cellular respiration is produced that way. But most of the ATP, 90% of the ATP that is generated during cellular respiration is produced by a different process called chemiosmosis and oxidative phosphorylation. And that goes back Well, we're running out of time. I'll come back to